And then what's really come in this most recent guidelines, and this has been the first attempt, I think, of trying to do what I would call patient-specific therapy, and that is that the CPP, should not be below 50, but this real range lies between 50 and 70. And why between 50 and 70? Well, again, it's difficult to do randomized controlled trials and TBI studies. So another way that you can do that is you can look at cohort studies to where patients are managed reasonably well between two different centers, but they have taken a particular perspective or approach to doing things. And so in Uppsala, as I told you, this is probably part of the whole low CPP crowd. They were actually trying to keep their CPPs above 60, their ICPs below 20. The Edinburgh crowd, by and large, was below 20, but they said below 25, and this was in the days of Rosner, and so they were going the CPPs greater than 70. So if you look, the patients were all about the same age, they were all, same sex, same type of injuries, so they were pretty well matched, except for the fact that, one group believed in high CPPs and one group believed in low CPPs. The good thing is that both groups were interested in measuring autoregulation. So they had a way where they could look at whether or not the autoregulation was intact. And by autoregulation, what I mean again is this was the curve I showed you before, which unlike other organ systems, as you increase the pressure, the CBF changed because of cerebral vascular resistance. Well, when this doesn't work, you become pressure passive. And so as the pressure goes up, you're pushing more pressure into the head. And what they found was that if you were autoregulating in Edinburgh, you did pretty good. But if you weren't autoregulating in Edinburgh, you didn't do so hot. And why is that? It's because you were pushing probably more pressure into the head, creating more ICP problems. Conversely, if you were in Uppsala and you were autoregulating, you didn't seem to do as well, but if you were not autoregulating, you seemed to do a little bit better. So how do you sort of apply that? Because in your ICU, you actually have both patients. And what this has allowed us to do is we've become much more aware of whether or not patients are autoregulated or not. So when the ICP is elevated, we don't just say give mannitol, we don't say just drain CSF. Particularly in younger patients where this is much more common to lose your autoregulation, we say, is the patient autoregulating or not? And if they're not autoregulating, we drop the CPP down. If they're autoregulating, we keep it up towards 70. So we're trying to reproduce this. And the bottom line is that one size just doesn't fit all. And we really have to get back to this idea of how are we going to do personalized care. So with these current endpoints here of the scan and the ICP and the CPP, I think we can begin to add things like brain tissue oxygen, SJVO2, in some people's hands microdialysis, and this assessment of autoregulation. So I just want to take you through a couple of cases here just to sort of illustrate how we use this. So this is a patient. We've got a good FiO2 response. This is what we typically do twice a day to make sure the probe's working. So we've got a 34-year-old guy, has a fall, seizure, GCS 11 on arrival, goes to the OR, comes back. The initial brain tissue oxygen tension is 6. It should be around 15. For those of you who work in places that use some of these protocols to say keep it above 20, this should be a concerning number. It was concerning to us. We did a challenge, we got it up to 23, so the device itself seemed to be at least somewhat responsive. And the ICP looked good, the CPP looked good, the SJVO2 looked good. So are we going to flog this patient to death because this one number's low? No, I mean what we did was we went down and we took a look and we said, well, we're not going to increase the FiO2 because everything looks good. The mannitol, no. Are we going to change the ventilation rate? No. Well, let's get a repeat CT scan. And so what we saw here, this is the first scan here, this is the second scan here. As you can see, the probe's going one way on this cut, and a little bit lower it's going the other way. And the bottom line is that this probe had actually not entered the brain. It was on the surface of the brain. And the only reason why I show you this is that it's good to look at numbers. It's good to respond to this and put something together. But your numbers are only as good as where your devices are and to make sure your devices work. So I show this to you so that you never place too much on one single number. And you need to make sure that you assess this before you start doing things to patients. So this is another patient here, 18-year-old sports injury, I think it was skateboard, GCS 6 on arrival, has diffuse brain swelling. We're of course going to monitor this patient. And if you're at our place, you'll get lots of monitors. You see there's an ICP monitor, that's a ventricular catheter, there's a brain tissue oxygen monitor, there's a CBF probe. And now, after about 12 hours, everything's fine, but the ICP is 34. So what do you want to do? Well, you're probably not going to sit there with an ICP of 34. And if you've got these numbers, and this was a real case, because the resident called me and said, well, the ICP is 34, what should we do? And I said, well, you know, we put all this stuff in this person's head for a reason, tell me the rest of the numbers. So this is the brain tissue oxygen, which was 67, so that was high. SjVO2 is high. The CBF is high. And the PaCO2 is a little high. So I know this was sort of a whirlwind tour of cerebral physiology, but what do you think is going on? Do you think this is a brain swelling problem? Could be, but probably not from water, right? Do you want to increase the FiO2 to make that number go up? No, the number's already high. Do you want to give mannitol? You think there's brain swelling? Probably not. Do you want to increase the ventilation rate? I mean, maybe. PaCO2 is 42. Do you want to get a CT scan? Well, see, I think actually if you understand the physiology and you understand how to use these numbers, what you're seeing here is a picture of hyperemia. The brain tissue oxygen's up, because remember I said that's proportional to the CBF. The CBF itself is up. The SjVO2 is higher than normal because the flow is going so fast, you don't have to offload that much oxygen off the hemoglobin. And so in fact what we did was we just took the PaCO2 from 42 down to 36 and brought the ICP down to 16. So again, we didn't give mannitol, we didn't take the patient down there. This is complicated. You've got to pay attention to each patient and treat them a little bit differently. I'll show you one more case and then I'll skip on over. Actually, let me just skip through this case because I do want to show you a little bit of the informatics work as well. So here's a patient here, 42-year-old female, status post-gunshot wound to the head, was 12 at the scene, declined to 6 in the ED, diffuse brain swelling. Of course, we're going to monitor them. This is what the scan looks like here. So ICP is controlled. We've got intermittent CSF drainage. Everything's fine. ICP now becomes 28 to 38. What do you want to do? Well, basically, I got a call from the resident who had already given 2 doses of mannitol reflexively, trying to treat this patient, saying, oh, well, the ICP's up, let's just give some mannitol. And so obviously, we're not going to give any more mannitol because it didn't work. And then I said, well, what else have you tried? Oh, well, after we did that, we tried some hypertonic saline. And that didn't work either. And so I was like, okay, so you tried 2 different types of hyperosmolar fluid. What else could be going on? Well, I said, again, let's look at the numbers. And so you see that the SjVO2 is a little elevated. The brain tissue oxygen tension is slightly a little bit high. The CBF seems to be sort of okay. The CPP is high. Is trying to do some of this directed care of having goal-oriented treatment, such as making sure that your PaCO2 is in the normal range, your PaO2 is in the normal range. And oftentimes we don't do this. I mean, it's strange if you go into an ICU and you see that the CO2 is all over the place. We let numbers fly here and there and we say, well, because the patient's sick. And what we found is actually just the opposite, is that if you try to put these patients into more of a normal category, you actually identify the outliers much more quickly than you would otherwise. And at least for us, this has helped us to improve how we're taking care of these patients. So to switch gears for a little bit for about the last 10 or 15 minutes here, how do we start to put all this together? Well, we certainly have seen that this idea of what most people talk about, bench-to-bedside research, where we're trying to go from a basic to a clinical environment, we're trying to develop drugs and markers and devices. We've used a lot of biomarkers in bioinformatics to study this. However, at the end of the day, what we're probably most interested in is how are we going to improve the outcome of these patients? How do we get this out to populations? And a lot of how you work in these larger environments is really through medical informatics, and herein lies the problem. So I still do a lot of molecular biology and work with a lot of folks, and we do genomics, and proteomics, and metabolomics, and all the omics. But if you look, we have so many cool tools out here for genomics. I have a chip right now that I could take a drop of your blood and I could sequence about 675,000 polymorphisms in you, boom, just like that. Amazing. What do you do with all this information? So we've got lots of tools out there and lots of ways of looking at this. And if we look over here where we are with medical informatics, I mean, we really are this Quonset hut over here. And why do I say that? Well, people said, well, this is a really data-intense environment, very, very complicated. It's too complex. This is a little kid here on ECMO. Look at all of this information we're getting. Now, for those of you who are in nursing and so on, you realize this stuff's important. I mean, we don't have these devices just because we're trying to help out the medical device industry. We know that we actually need to monitor some of this stuff to keep people alive. But what do we do with all this information? What we do is we get a really highly skilled and trained nurse to basically stand at the bedside to look at all this information. We have a whole team of physicians that sort of look at this information, and this is our computer. We're processing all this in our head. And so, you know, if we look today, we know that all this physiology is important, but what do we do with all this data? In 80% of the ICUs in this country today, this is what we do. We put it on a 2-foot-long sheet of paper. Okay, this is the same format of paper that Harvey Cushing first laid out when he was doing an anesthesiology rotation at the Massachusetts General Hospital. I mean, for 100 years we've been extracting this kind of information. It gets worse. I mean, this is a rounding scene from a typical hospital. You're walking around, all the little docs are there, they're talking to the nurse, and I want to draw your attention to what this guy's holding right here. This is medical informatics in 2011 for most places. I was over at the hospital this morning. It blows me away, you know, that I have an iPhone 4 here, that I can do all this stuff, and then I've got a set of 3x5 cards in this pocket over here. There's something wrong with this picture, right? We've got to have a better way of looking at this. Because you know what it does is it really changes the way that we treat people. I mean, if you can't harness all this information, then you have to start basically trying to do things one step at a time. But we know that there's 5 things going on at the same time. And so to use my friend Claude Hemphill's quote, you know, we treat univariately in a multivariate world because we can't capture and process this information. It really changes the way that we practice medicine. So I don't think this is good. I mean, we have issues with pharmacokinetics. We have issues with multiple regression to have better models for disease prediction. You know, here on one hand in cancer, you know, we can separate acute lymphoblastic leukemia from acute myeloblastic leukemia with a gene chip. And then here we're still on mild, moderate, severe. So I think, you know, we've got to get back to processing this stuff a little bit better. So, you know, for the last 8 years, we've been struggling to build a better informatics system in the critical care arena for traumatic brain injury so that we could try to do better diagnosis. We could try to understand these physiological abnormalities because I do believe even in areas with less resources, if we had some clinical decision support tools, some of those complicated cases that I was just running through, I mean, there's nothing magical about what we're doing. We just understand cerebral physiology and interpret the data. So we should be able to have some computer decision support to be able to do that. And that's really been sort of the goal. We've been fortunate to have some good collaborators here in the area. Intel, which is just right up the road here in Santa Clara, we worked with their digital health group and our computer science colleagues at Cal. And one of the problems has been just simply harnessing this information. Not one of these devices speaks to each other. So we could go to Fry's Electronics and I could buy a printer, I could buy all kinds of stuff, I can plug it right into this thing. In fact, I walked right up here and plugged my Apple into this PC device. Why? Because there's communication standards. There are no communication standards in the medical device industry. Not one of these devices talks to one another. In fact, many of them are made not to talk to one another. And so it took us about 6 years to build a reliable, robust data acquisition system that we felt comfortable using in the clinical arena so that we knew that we were capturing this data. So what do we have now? We have a lot of bedside physiology. I do believe in molecular biology, so we are collecting DNA, we're collecting serum samples, we have an imagery repository, and this sort of infrastructure was what we used to do this prospective TBI trial that we're now embarked on right now. So let's go back to our medical informatics card here and look at something like Tmax of 39.4. Well, what does this mean? Does this mean that the guy was febrile all night? Was this just a single abnormal number like one of those other abnormalities I showed you? We started thinking about, well, how about the fever burden? It would make sense to me that if you had a fever for 4 hours, you might be sicker than if you had a fever for 2 minutes. And this is an example from some work that Claude Hempill looked at, looking at patients with intracerebral hematomas. And what you can see, this burden of fever between what we thought was kind of a zone where we were worried, but we never did anything until the fever popped up to 38.5. Turns out that this fever burden is very predictive of outcome. Now, is that cause and effect? Probably not. It just means that when you have the fever, you probably have some inflammation. And inflammation in the setting of ICH probably is a bad thing. But it also shows you just how easily you can start to look at some of this stuff, but you can't do that unless you can process things. I mean, another way of looking at it is that we're trying to run a business, you know, using receipts versus Excel. You just can't really do very well. So we've shown over the years that this relates to blood pressure, it relates to glucose. We've seen that the burden or dose of ICP is important for outcome. We've also gone back and looked at observational data where if you've got a change in practice, so, you know, or not a change, but a difference in practice, there's no guidelines for how much mannitol to give patients. And some clinicians like to use 50 grams, some clinicians like to use 100 grams. So you can use that sort of as an experiment as long as these populations are reasonably well matched. And you can actually find out things like if you give more mannitol, it works for a longer period of time. And this may seem very obvious to you, but this was the first report in the literature using a relatively large patient series that came out of just some of our data acquisition that this is in fact the case. So really what we're trying to do today is working in between these two domains of data mining. And one of them is knowledge driven, which says that you can build a model and then you work off the model. The other way of looking at this says it has to be data driven. You really don't understand how the body works. It's too complicated. You need to process the information. And in medicine, we work somewhere in between. You know, I have models of physiology, like we've been talking about this afternoon. I then look at the data, and I look at patterns of the data, and I try to mesh those two things together to make them fit. So I think if we're going to make clinical decision support tools, it has to be somewhere in the middle. So the other problem that we have is just how do you, even if you acquire this data, how do you visualize it? So this is 36 days of continuous physiological data from a patient, and it represents about 4 million data points. You can't even take this and put this on your computer with standard software. So we're writing software to be able to even visualize this data. Is it important? Yeah, you know, I won't go into this because we don't have time today, but there are patterns in this, there are things that you can do, but we have a really long way to go to get off the 3x5 cards. So I'll leave you with just one example, which what I hope will be practical and you can take away from. I've just got a few more slides here. So one of the problems that we have is alarm fatigue. So alarm fatigue is a result of the fact that 86%, this was the most recent paper, of ICU alarms are false positive. So if 86% of the time that it's false positive, and you're a bedside nurse, who here pays attention to the alarms when they go off? Who here turns them off? You know, most people do, and it's called, I mean, it's well known, it's called alarm fatigue. So people have tried all kinds of ways to fix alarms. So they've used rules-based systems, they've used running averages, they've used all, none of it's really worked very well. And so one of the things I've been working on with my colleague at Cal, Stuart Russell, who's really a world's leader in artificial intelligence, is how can we make smarter systems, not only to look at this physiology, but even taking something as simple as how could you filter out an alarm? Well, the first thing that you have to do is you have to know how this thing works. And I actually thought I knew how these things work because Stuart asked me, he said, you know, well, we're looking at all this data. Why do you think you're getting all these artifacts? And I was like, well, I think, you know, the transducer moves around. I know that they open it up to draw blood gases and it goes down. He goes, yeah, but how does it work? And I said, well, the way it works is that you've got this pressure transducer. There's an arterial line that goes in there, and then you measure the pressure on the monitor. And we were in the ICU, and he said, what's that bag for, that pressure bag for? And for those of you in nursing, you realize, you know, doctors don't know how to use those pumps. We can't turn them on, we can't turn them off. And clearly I had no idea what the hell this pressure bag was for because I've been looking at it, you know, for 15, 20 years and never paid attention to it. She goes, oh, no, that's part of the system because you've got to be able to have back pressure on this line because it's an arterial line. And so Stuart says, well, what do you usually pressurize that thing to? And she goes, oh, usually between about 250 and 300. He goes, you know, a lot of your artifacts are between 250 and 300. And I think what this shows you is the magic of working with somebody that's completely out of your field, that's very smart, that comes in and asks questions that clearly you didn't know the answer to or would have never even thought to ask in the first place. So from this, then comes this. Don't pay attention to this, it's too complicated. But what this is a probabilistic model that says, okay, we're going to like try to model the human mind. So where if you walked in and you saw an artifact, what are the things that you would think about? And then you can start building probability distributions of this. It's kind of the way speech recognition works. You know, it's not trying to recognize every pattern. It's listening to words and trying to look at the probabilities of what the next words are. And so it uses that combination to match that up to make it work as well as it does today. Well, does it work here? I won't go through this in detail other than to say that this is what it thinks the bag pressure is here on the left until it starts seeing some numbers. And once it sees numbers, it can start to refine this. So you can take these models and you can train them. So we took this model, this very complicated model back here, and we trained this on about 300 patients, I think. And so we taught it how to recognize all these artifacts. And, you know, we started to be able to make estimates of what the bag pressure was and so on. And so from an informatics thing, it was really, really cool. You know, from my practical perspective, I'm like, okay, so this is really neat that you do this. Does this thing actually work? And so the way that you determine whether or not it works is that you basically start showing it patients that it's never seen before. And so this is a patient that this algorithm has never seen before. It's got those spiking artifacts. It's got these artifacts that go down low. And so when you run it through the algorithm, what you see is that you basically almost clean this up. And when you go through and you look at a number of patients, we can reduce the false positive rate to less than about 10%. So to me, I think this is an early example of how, working in a multidisciplinary group, even trying to look at something as simple as alarming, because, to say that I'm going to build a clinical decision support tool, I'm not going to just come out there and put something together. Somebody's going to say, oh, we're going to give mannitol, we're going to drain CSF, we're going to take them to the CT scanner. We've got to have more sort of tangible goals such as this. So I'm not going to really show this because I'm almost out of time. I just got the 5-minute signal there. But what I would like to say is that, in general, I think that we need to take a broader view of what we're looking at this. We need better informatics. We need a lot of these devices, that we're doing over here in this T1 translational point. We need to continue to push that. But somehow we need to be able to mesh all this together so that not only can we make these transitions across T1, but also get these devices and ways of looking at this out in the population and having better tools to do that. So lots of folks to thank. I'm really only the sum of all the wonderful people that I have working with me in the neurosurgery department, neurology, surgery, informatics, computer science at Berkeley, the folks at Intel. And I'll stop and take any questions there. Thank you very much.